AgCenter Mendel University Food Symposium, where today we're going to talk about the interplay of nutrition and immunity in the human body. My name is Sarah Bernard, and I work with the LSU AgCenter's Global Network. It's my honor this morning or this afternoon to introduce our moderator, Dr. Daniel Falta, who is an associate professor of animal breeding at Mendel University. Dr. Falta is Vice Dean for International Cooperation at the Faculty of Agri-Sciences, and he has been essential to the LSU Mendel partnership since its inception. Again, welcome to all, and welcome to Dr. Falta.
Thank you for introduction of myself, of my person. Thank you very much. Good morning in America, good afternoon in the Czech Republic. Dear ladies and gentlemen, dear guests, attendees of today's Food Symposium event, which runs from Monday until Friday, as you know. Thanks to the organizers who trusting me and set it up me as a moderator of today's section. In the introduction part now, let me briefly introduce today's presenters, who will be that 5 persons. They will be Dr. Rebello, Dr. Chen, and Dr. May from LSU, and then Dr. Matejovičová and Dr. Komprda from the Mendel University. Today, again, you will see that the topic of nutrition and human health, especially in the COVID time, is very interesting because each of us has to eat theoretically every day. Of course, somebody says that it's possible to not eat for 3 weeks. So maybe you will learn something more and you can discuss with our speakers who will present. So I would like to challenge you to give questions to the presenters via chat. So thank you very much for that time at the beginning. And I would like to give the online space to the first speaker, who will be Dr. Matejovičová. So Dr. Milena, Teams is yours.
Okay, thank you very much for possibility to present my presentation. First I will ask, could you hear me?
Yes, we can hear you.
Okay, thank you very much. Firstly, I want to say hello to Louisiana and good afternoon to Brno. And I will find my presentation first. Well, so this is the first time I have possibility to present online, so I'm a little bit nervous. Okay, I hope it's visible.
We can see your presentation.
Okay, then let me present the presentation with the name The Potential of Nutrition in the Prevention of Chronic Civilization Diseases as a Possible Protection against the disease COVID-19 complication. It's summarizing data from literature which are known about the metabolic diseases and their interaction with the disease COVID-19. First, I was challenged to present something about myself. So I work as a assistant professor at the Department of Food Technology. I'm educated in biology, and my specialization is medical biochemistry. So I have practice in teaching medical chemistry and biochemistry for nearly 20 years. At first, I was employed at the Faculty of Medicine in Slovakia, and further I have taught medical chemistry at the Medical Faculty of Masaryk University in Brno. So my scientific interests are biochemical mechanisms of various pathogenesis with the precipitation of oxidative stress. And also I'm interested in regulatory effects of nutrients in different states of health and disease. So my responsibility here in Brno at the Department of Food Technology are specialized courses in human nutrition, such as preventive nutrition, psychology nutrition, community nutrition, and diet recommendation. So let's start with my topic. So long before the COVID-19 pandemic appeared, there was emerging pandemic spreading about several non-communicable chronic diseases, which are called also civilization diseases. This group of diseases has multifactorial causes. There are exogenous causes that are mostly connected with the lifestyle, which is common in developed countries, and there are endogenous causes which are connected mostly with the genetic background of the organism. And there is strong association with the Western diet. This diet is common in developed countries, and it has atherogenic potential. It's rich in saturated fats, refined carbohydrates, low unsaturated fats, with unfavorable proportion of N-6 to N-3 polyunsaturated fatty acids, and it's also pure in fiber and antioxidants. So the major diseases are these four highlighted non-communicable diseases, which are responsible for major proportion of deaths globally. According to World Health Organization, the number one globally in cause of deaths is cardiovascular diseases, the first one. Then cancer, diabetes, and chronic respiratory diseases. The cardiovascular diseases are responsible for death of 17.9 million people per year, which is over 30% of all deaths globally. So it's together all these highlighted chronic diseases are responsible for more than 70% of all deaths globally. Cardiovascular diseases are connected with those medical states which are hypertension, dyslipidemia, obesity, and resistance to insulin. These risk factors, medical states of the organism are going together, they are connected together, and not only cardiovascular diseases, but also the other diseases, for example, the diabetes and cancer are linked to some of them. So mortality has trends to decrease last years in developed countries, but the prevalence is still high. The mortality decreases because there is better management of diseases, and this, for example, in cardiovascular diseases, increased lifespan, but there is low quality of the life in such people. Trends for future, there are plans to manage these non-communicable diseases, which are planned to decrease by 30% till 2030 a year. But those are plans according to the Health Organization, and we will see if it's come to true. I will concern to obesity in my presentation because obesity is common to nearly all of this group, all of these diseases of this group. Obesity is defined as abnormal or excessive fat accumulation that presents a risk to health, according to World Health Organization. It's characterized by body mass index higher than 30 kilograms per meter square. It means this is the proportion of body weight to body height. There is a risk with visceral obesity, which is connected with chronic inflammation, oxidation stress, insulin resistance, and this is connected with high fasting glucose, then hypertension, high blood pressure, and dyslipidemia. Dyslipidemia is specific with high triacylglycerol and low HDL cholesterol in this type of obesity. If there are three of those four medical states, which means visceral obesity, insulin resistance, hypertension, and either high triacylglycerols or low HDL cholesterols. If there are three of them fulfilled, we can speak about metabolic syndrome. Metabolic syndrome leads to endothelial dysfunction and further to atherosclerosis and heart diseases, and its potential of atherogenic diet to induce this dysfunction also, and on the other hand, also atherogenic diet increases the fat accumulation and the visceral obesity also. Next slide shows adipose tissue functions which are mostly impaired in obese people. Adipose tissue has primarily storage function. It's considered being organ with various functions, but they can be all impaired in obesity, as a consequence then obesity, and adipose tissue consists from several types of cells. That most abundant are adipocytes. We can find adipocytes of two types. There are white fat, which can be divided to peripheral and visceral. This visceral within the abdominal part of the body is the most important for the metabolic syndrome, and there is brown fat, which morphologically differs. The cells, the adipocytes, differs from the white fat. In the white fat, there is one big fat droplet and sparse mitochondria, and in brown fat there is much more mitochondria and small droplets, several smaller droplets of fat. What happens when there is much more fat intake than can be stored in the adipocytes? Normally, if the capacity of the adipocyte is exceeded, the cells, the pre-adipocytes, can differentiate to the adipocytes. This is impaired in fat people, in the obese people. Then when the capacity of the adipocyte is exceeded and there is no more space for fat, the hormone activated lipase is activated even after meal postprandially, and fatty acids are released to the blood, and they are stored in the other organs and cause so-called lipotoxicity. So the other function of the adipose tissue is a secretory function. There are many molecules called adipocytokines that have hormone function, and other molecules which are secreted by other cells, for example, immune cells. But in obese people, in the adipocytes also can be expressed and secreted by adipocytes. There are adipokines which has function as for down regulation of insulin resistance, down regulation of the food intake, and they have preventive function, as like is, for example, leptin or adiponectin or omentin, and there are adipocytokines or adipokines which have function that they have pro insulin resistance function. They upregulate insulin resistance. For example, for with fattin, it binds to the insulin receptor at a different site than insulin and decrease insulin affinity to the binding site. So it has function in insulinoresistance, and it's expressed in higher amounts in the obese person. So the resistin, for example, then its expression increased also in obesity, and it's connected with insulinoresistance. Agouti-related peptide, it has also orexigenic signaling. It make signals for intake of food. It transfer the signal to brain. These other molecules have pro-inflammatory function, and they increase inflammation in the fat tissue. Then next slide shows the dysregulation in hormonal signaling which is processed by these molecules. In normally functioning signaling, there is during before meal, during hunger, orexigenic signaling, ghrelin in stomach, it activates in hypothalamus the center, which at the end increases the appetite and decreases the energy expenditure. After meal, there should work anorexigenic signaling, which are transferred by leptin or insulin, or gastrointestinal peptide cholecytokinin, which inhibit appetite, decreased appetite in hypothalamus, and they increase energy expenditure. Changes in nutritional state can modify this complex signaling. In obesity, this signaling, which signal anorexigenic state, is impaired. The gastrointestinal peptides leptin and insulin are resistant. The answer to these peptides is resistant, and normally, while the calories restricted states are present, there is no such resistance demonstrated. So this is impaired, and then energy uptake is higher than the energy expenditure. In obesity, there is chronic inflammation. As I said a few slides before, there is a lipotoxicity of the fat tissue. Fats are accumulated in other tissues. There are macrophages which are attracted to the adipose tissue, and also they can be differentiated from preadipocytes. It can be stimulated by pro-inflammatory cytokines like interleukin-6, tumor necrosis factor, and C-reactive protein. In the tissue, this fat tissue is oxidative stress taking place because there is no enough oxygen in the abundant hypertrophic adipocytes. So the adipose tissue is suffering from lack of oxygen. There is oxidation by this state of lipoproteins. The low-density lipoproteins particles are oxidated. Macrophages infiltrate the blood vessels, and they attack these oxidated particles because they are antigens for this for the immune systems, and should be removed from the blood. But this LDL lipoproteins particles oxidation leads to formation of atherosclerotic plaque in the vessels. In obesity, there is shown that there is activation of innate immune system, but the adaptive immune system is inhibited. So the T and B lymphocytes function is suppressed, and there is a bad or worse answer to the immune stimulus. What about the disease COVID-19? This was first identified at fall of 2019 in the China. We all knows the name of Wuhan. It's a respiratory infectious disease which is caused by beta coronavirus, and the virus was called because it's very similar to the SARS coronavirus, so it's called SARS-CoV-2 coronavirus. And symptoms of most patients are typically for the respiratory disease. This is cough, fever, myalgia, fatigue, and it's presumed that most of the cases are asymptomatic. Then they are without illness, without these symptoms. Why then it's so dangerous? This disease, because it's highly transmissible, and also there is some population of patients which are very vulnerable, which have some comorbidities, and they can develop severe cases, severe symptoms of the disease, and it can be finished by respiratory failure, septic shock, and by death of these patients. So the virus entry to the cells by a cellular receptor of the angiotensin II converting enzyme. This receptor is spread through the body, so the virus can enter different tissues. Then there is a major site of the entry in the respiratory tract, but it can be also in other, for example, gastrointestinal tract found, and it's connected with gastrointestinal problems and symptoms. What are risk factors from the literature? the risk factor can be divided by the different criteria. Demographic factors are the male sex and older age. It was shown that 70% hospitalized have age over 45 years. And further, there are comorbidities which are hypertension, diabetes, and heart diseases, and also obesity. These are comorbidities which are connected with higher mortality and with the severe progression of the disease. In Italy, Lombardy, there was a study among nearly 2,000 patients which were treated in the intensive care unit, and it was shown that 78% have at least one comorbidity, and all comorbidities are linked to the obesity. So it's not a small portion of patients. 70% of patients which needed intensive care were obese, or they have some other comorbidities. What is the outcome of obese patients with COVID-19? There is increased risk of hospital admission, according to literature. There is longer hospitalization of obese patients, approximately, according to different studies, approximately 5 days or 1 week from some cohort studies. There is nearly 2 times higher likelihood to be admitted to the intensive care units. This was found in USA, in New York hospital, and there is body mass in patients more than 30 kilograms per meter square, so it means they are all obese, and this is the twice factor. This twice increases likelihood to be hospitalized to the intensive care units. Most studies employed multivariate analysis, so it means that there were also obesity-related diseases taken in the attention, for example, cardiovascular disease and hypertension or diabetes. And obesity increases the risk of severe COVID-19 and death, including also young patients. So not only elderly, but also in young people, there is higher risk of severe progression of the disease. What was found in previous outbreaks from other coronaviruses in the last 2 decades, there were 2 outbreaks of coronaviruses caused diseases or respiratory diseases, and in all patients which suffers of those diseases, there was increased risk if they were obese. It was found also the higher risk factor in obesity in influenza, which was caused by virus H1N1. and also in other respiratory infections, the obesity is a high risk factor for severe progression of the disease. Here I dare to show a graphic from colleagues from China that clearly explain what happens with obese patient and how the complication rise in obese patient and what is the link between the complications. So obesity is connected with, for example, depletion of vitamin D. There is pro-inflammatory state. There is high expression in the respiratory tract of receptor for angiotensin 2 converting enzyme, and also there is compromised the immune function and the respiratory function. So the virus is recognized by immune system, and then the effectors T cells will cause lysis of the virus, and the immune system will overreact, and the reason this overreaction is the high synthesis and excretion of cytokines, which are excreted in a very high amount, which leads to the very severe reaction of the organism, to severe complications of this respiratory disease, and can lead to multi-organ problems and death. What was curious, what I found in some of the novel papers, it was that there is less severe cases in smokers and also in patients with some pre-existing respiratory disease than it was hoped before, and there are not reported in the recent later studies new cases with or higher incidence in among this group of patients. So smokers and patients with respiratory diseases are not in the higher risk than normal healthy people. In the progression of this disease seems to be important presence of some metabolic disorder, which included dietary lifestyle and is represented by glycemic disorder, lipid disorder, obesity, and also sedentary lifestyle, and also the other severity modifiers, of course, such as high blood pressure, hypertension, high age, and also inflammation, which aggravate the COVID-19 induced organ failure. So, which can be recommended that aim to decrease risk factors for COVID-19 disease is decrease the risk factors and boost immune function. Then can be advised to decrease energy intake, so the body weight decrease by just 10% can decrease the risk of cardiovascular disease, and also hopefully the risk of the severe COVID-19 progression, and also the risk of the obesity decreases insulin resistance, the decrease of obesity by 10%. There should be a blood glucose control. So complex carbohydrate, it's in the food should be recommended. The low glycemic load and low glycemic index food, and also the lowering unsaturated fats and increase N-3 polyunsaturated fatty acids, which can be found in fat, fishes, seeds, and nuts. And there is recommendation to consume high quality protein and include some fiber sources, which is increased fruit and vegetable consumption, and also some antioxidants, which are found also in those commodities, fruit and vegetables. There is also a recommendation to lower salt intake, which is connected with prevention of high blood pressure, with hypertension. This fulfills some types of diet, so-called DASH diet, which is formed on the basis of Mediterranean diet, and this is the approach to stop hypertension. It's low in salt and high in monounsaturated fatty acid and polyunsaturated acid, and also rich in vegetables and fruits, whole grains, and low-fat dairy products. In the protein sources are found poultry and fishes, and also nuts, and also some beans and legumes, and non-tropical vegetable oils. So this diet is recommended as prevention for cardiovascular diseases, for high blood pressure, and also in restricted calories regime, it will have a positive impact on the obesity, on the decrease of body weight, and hopefully this can prevent severe outcome of COVID-19 disease. So here is some literature sources which I used, and thank you for your attention and for patience. I didn't keep my time, but so it's my problem, sorry.
Thank you, Dr. Mateovičová, for your very nice and interesting presentation. Maybe a little bit warning, especially for obese people. As we know, I cannot see any questions in the chat, and we had also some technical problems with Microsoft Teams. Maybe I will use it, let's say, to save a little bit of time, and we can leave the questions on the discussion, let's say, at the end of today's block, because maybe we will have a much more information if you agree. Our presenters also can join to the discussions with a microphone. And second speaker, so thank you again, Dr. Matejovičová.
Thank you also.
Welcome. I can read a message from Ivana. Too much of good food in the nutrition recommendation. Is there too much of good food in the nutrition recommendation? We should be aware. So you have a question, if you can see it in the chat.
Yes. I don't know how I have to understand too much of good food. Of course, the first we have to take in account is the calorie intake. We don't have to exceed the calorie intake, and it's better to avoid very fat or energy rich food, and instead of this eat some low energy and high nutrition food like is vegetable and fruit. If vegetable and fruit will make 1/3 of our energy intake, it will be very nice, and the calorie intake will not exceed the need of the body. So that's maybe this. Is it okay like that, the answer? I don't know if this was the point of the question.
Yes, thank you.
Okay, thanks.
Thank you. And but for example, in the undeveloped countries, how they can have access to this high quality proteins?
Well, this is a worldwide problem that most of the people at the earth, they don't have access to high quality protein, and this was also mentioned by World Health Organization, that this is main aim to find ways how to decrease vulnerability of these people who don't have the access to the protein. So that's a known problem, but it's not so easy to solve this problem.
On the other hand, we can see in the chat a comment from Prof. Komprda that we have let's say at the Western countries or well-developed countries with a highly processed foods. So contra the countries who are undeveloped or less developed. Thank you very much. And second speaker for today will be Dr. Tomáš Komprda with his presentation about selected micronutrients and nutraceuticals, what means ingredients that have a beneficial effect on our health, maybe. So Tomáš, Teams is yours and you can upload your presentation.
Thank you, Danny, very much. I'm trying to find my presentation.
Yes, we can see it.
Okay. Once more, thank you very much, Danny. Good morning or good afternoon, ladies and gentlemen, dear colleagues. Allow me to submit my presentation about selected micronutrients or nutraceuticals as possible immunomodulators in COVID-19 prevention. First of all, it's our duty to present background about ourselves. So regarding my background, I have a master's degree in chemistry and a diploma of veterinary medicine and PhD in animal physiology. Currently, I am member of staff of Faculty of Agri-Sciences, Institute of Food Technology, and I am a full professor here. My current interests in research are long-chain polyunsaturated fatty acids and -3, but I would like to underline that it is not in connection with COVID-19. It's not my first level of knowledge in this connection. Regarding my teaching duties, I am responsible for a freshly introduced study program, Food Sciences and Human Nutrition. And within this program, I am teaching subjects human nutrition, functional foods, and food toxicology. So now I would like to proceed with this my topic. I have divided my presentation to two parts. In the first part, I will comment a relationship between SARS-CoV-2 and the immune system. And in the second one, I will mention some food components whose intake can contribute to decreasing the probability of contracting COVID-19. What is the relationship between immune system and effects of SARS-CoV-2? As it is very well known, SARS-CoV-2 enters the cell in this context. It's type 2 pneumocyte via angiotensin converting enzyme 2, but it's rather strange that after entry, this virus downregulates expression of this receptor. And this leads to disturbed ratio of angiotensin 1 to angiotensin 1,7 with a consequence of pulmonary edema, inflammation, and so-called cytokine storm. Despite this cytokine storm, the most important defensive cytokine, interferon alpha or interferon gamma, are decreased as an effect of SARS-CoV-2. It's very interesting. Regarding immune competent cells, I would like to mention T cells, neutrophils, and monocytes. Regarding T leukocytes, there are, as you know, two types of T leukocytes, helper T cells, TH, and cytotoxic T cells. When SARS-CoV-2 enters the cell via toll-like receptor, this helper T cell is transformed to Th1 mode, which means it's in inflammatory modus with already mentioned cytokine storm. The main pathology regarding neutrophil action is formation of so-called neutrophil extracellular traps with a consequence of platelet aggregation and thrombosis. After activation of monocytes, in these cells, the signaling pathway of nuclear factor kappa B is activated, which leads to skewed inflammatory cytokine production, including interleukin-6. And moreover, monocyte-derived macrophages are pressed to infiltrate lung tissues, which further worsens the situation. As you can see, in damaging inflammatory response in macrophages, there is a leakage of blood vessels, and monocytes and macrophages can easily enter alveolus. Regarding neutrophils, this is also only an illustration of these neutrophil traps, because these extracellular neutrophil traps are causing blood clotting. It's a very severe complication of SARS-CoV-2 infection. In one previous slide, I showed you T cells, Th cells, helper T cells. Here we can see effect of SARS-CoV-2 on cytotoxic T cells. And it is very interesting and strange that these T cells seems to disappear. And the reason for this disappearing is until now unknown. And it's also worsening the situation with COVID-19, because these cells, cytotoxic T cells, are able to destroy viruses, including SARS-CoV-2, or cells infected with these viruses. As far as second part of my presentation is concerned, I would like to present some words about nutrients that possibly could decrease risk of severe outcome of COVID-19. As you can see, all vitamins are possible to positively influence immune system. Because we have a shortage of time, I would like to chose only vitamin D as immunomodulator and antimicrobial substance, and within minerals I would mention zinc as antioxidant and substance which is able to halt virus replication. Within the plethora of nutraceuticals, which are an ability to prevent COVID-19 is being attributed to, I chose only two examples, palmitoyl ethanoyl amide and long-chain polyunsaturated fatty acids N-3. As I have already mentioned, in our laboratory, we are dealing with these fatty acids, but not in connection with COVID-19. Nevertheless, also these fatty acids, I think, have something to say as a prevention of COVID-19. Let's start with vitamin D that helps destroy viruses, including possibly SARS-CoV-2. This coronavirus, via its pathogen-associated molecular patterns, enters toll-like receptors in immune cells, but in consequence of this activation of the immune cells is increased expression of vitamin D receptor, because it is necessary to underline that vitamin D is actually a hormone, and after recruiting its receptor with vitamin D receptor, this vitamin D receptor binds second receptor, retinoid X receptor, and this dimer in the cell nucleus acts as a transcription factor for expression genes coding for among others, two important proteins, namely catalycidin, which is able to destroy viral envelopes, and as it is known, SARS-CoV-2 is enveloped virus, and the second protein is it's a group of proteins, so-called defensins, that are able to act as chemoattractants for inflammatory cells that are involved in the immune system, in the defense immune system. Moreover, vitamin D improves the action of interferon gamma. As I have already mentioned, these interferons, alpha or gamma, their concentration is severely decreased in COVID-19 situation. So improving action of these cytokines, it's very positive in this context. As far as other actions of vitamin D are concerned, with vitamin D is, I mean vitamin D3, is able to increase expression of angiotensin converting enzymes 2 in the lungs. as we already saw in previous slides, in COVID-19 infection, despite this virus enters the lung cells via angiotensin converting enzyme 2, concurrently the virus decreases expression of this receptor. So increasing expression of ACE2 is one of preventive measures. I mean preventing of the pulmonary vasoconstriction response to SARS-CoV-2, and it leads to reduction of the lung injury. Vitamin D also stimulates surfactant synthesis in alveolar type 2 cells, and concurrently helps regenerate endothelial lining in lungs, and this attenuates alveolar damage and also decreases the risk of pulmonary fibrosis, which is one of very important negative outcomes of SARS-CoV-2 infection. Viruses, including SARS-CoV-2, are able to disturb cell junction integrity. Vitamin D contradicts this action because vitamin D3 maintains tight junctions in proper state, including gap junctions and adherent junctions. Moreover, vitamin D3 also weakens the cytokine storm for, among others, following mechanisms. Vitamin D3 represses production of interleukin 2, and this action leads to suppression of the Th1 responses. We already saw that transformation of naive T helper cells to the mode of Th1 is deleterious in this context. So suppression of the Th1 responses is very positive action of vitamin D3, and moreover, vitamin D3 promotes contrary response, that is T helper cell 2 mode. So instead of deepening pro-inflammatory state, I would say calming down situation is preferred. Vitamin D3 also inhibits the signaling pathway of nuclear factor kappa B, and as a consequence decreases expression of its pro-inflammatory target genes. Because vitamin D3 increases gene expression of several antioxidative enzymes, including glutathione reductase, it is able to counterbalance oxidative stress. Despite my following presentation of vitamin D. Currently, it's necessary to admit that currently there is no clear epidemiological evidence that vitamin D could significantly impact the COVID-19 incidence or its severity or mortality. But on the other hand, unequivocally we can call vitamin D a preventive micronutrient. I would like to say something about recommended daily intake of vitamin D, but it is also necessary to mention that there in the nutrition society there is some controversy regarding what's the normal or optimal serum vitamin D level. Nevertheless, I would recommend this value approximately 10 micrograms per day. As a preventive intake in current pandemic situation, some authors recommend as high level as 125 micrograms vitamin D per day. The most important source for human cells of vitamin D is ultraviolet radiation, and where vitamin D is formed step by step in skin, then some transformation is performed in the liver and finally in kidney. Regarding food sources, the most abundant vitamin D3 source is fish oil that can deliver 210 micrograms per 100 grams. So 1 tablespoon contains approximately 35 micrograms of the vitamin. Also fish fillet, 100 grams of the fish fillet is able to deliver 7 micrograms of vitamin D. Regarding the further nutrients, from the group of minerals, I have selected zinc for its ability to inhibit viral replication through inhibition of viral RNA polymerase activity. I'm not able to count how many times I have already mentioned interferon alpha and interferon gamma in the connection that these cytokines are decreased in COVID-19 infection. So it is worthy to mention that dietary zinc is able to induce production of these interferons by activated T lymphocytes. And apart from it, zinc also enhances the antiviral action of interferon, especially interferon alpha. It has been found, demonstrated that zinc, via the increasing the interferon gamma, increases also concentration or activity of zinc-based metalloproteases and interferon-induced transmembrane proteins, and that means important antiviral activity against, in general, against enveloped viruses, and as I have already mentioned, SARS-CoV-2 is also or belongs to the group of enveloped viruses. As far as recommended intake of zinc is concerned, US Institute of Medicine recommends there are two stages how to evolve recommended dietary allowance. First step is so-called estimated average requirement, and this is for women and men approximately 7 and slightly more than 9 micrograms per day, and corresponding recommended dietary allowance is 8 and 11 micrograms per day. The most important food sources of zinc are, for instance, beef, pork, also poultry meat, milk and milk products, and also full corn cereals. Regarding zinc, and in the context of COVID-19 pandemics, it is worthy to mention some risks of supplementation with zinc, because there is a relatively small difference between what is recommended and what is already toxic. This gap is relatively narrow. So I would recommend to consume sufficient amounts of meat. I have mentioned beef, pork, poultry meat, whole corn cereals, milk and milk products, but I would be very careful regarding supplementation. Let's proceed to nutraceuticals. I chose two examples. First one is palmitoyl ethanol amide. The reason for this selection is its structural resemblance with classical endocannabinoids, despite the fact that this substance has no activity on cannabinoid receptors. But nevertheless, palmitoyl ethanol amide has analgesic, anti-allergic, and most importantly, anti-inflammatory activities, including inhibition of mast cell degradation, and acts via selective activation of peroxisome proliferator activated receptor alpha, which antagonizes nuclear factor kappa B signaling pathway. Also, it's worthy to mention that palmitoyl amide reduces formation of reactive oxygen species, and so it's able to prevent endothelial damage, including damage in the lung tissue. Last but not least, I would like to mention long-chain polyunsaturated fatty acids N-3. It is known that SARS-CoV-2 is able to reprogram metabolically the host cells via binding to via activation of sterol response element binding protein, and long-chain polyunsaturated fatty acids N-3 are inhibitors of this sterol response element binding protein transcription. So long-chain polyunsaturated fatty acids N-3 can counteract the activating effect of the virus. I chose as an example one of this group, from this group of fatty acids, docosahexaenoic acid, that is natural ligand of peroxisome proliferator activated receptor alpha. This receptor is activated by DHA, enters the cell nucleus, and binds to promoter of the gene coding for so-called INSIG protein, and this protein is able to sequester sterol response element binding protein 2 in cytoplasm, and the signaling pathway to the of this sterol response element binding protein, its signaling pathway to the cell nucleus is interrupted. Long-chain polyunsaturated fatty acids N-3 are also able to inhibit the signaling pathway of nuclear factor kappa B, and in this sense are inflammation modulators. When we have in our foods plenty of polyunsaturated fatty acids N-3, these fatty acids are enriched in the plasma membrane, and after releasing from the membrane, docosahexaenoic acid binds fatty acid binding protein, and consequently binds peroxisome proliferator activated receptor. This receptor is active and enters the cell nucleus, and with coordination, in coordination with retinoid X receptor, acts as transcription factor of the gene coding for protein that is inhibitory protein for nuclear factor kappa B. Nuclear factor kappa B is here shown as dimer of two proteins, P50 and P65, and this nuclear factor kappa B is activated by the action of formerly mentioned interleukin 6. We already know that so-called skewed signaling of macrophages leads to, among others, to increase of plasma concentration of interleukin 6. After binding to its receptor, the signaling pathway of nuclear factor kappa B is activated, and this nuclear factor enters the nucleus, binds to promoters of the genes coding for pro-inflammatory cytokines, for instance, tumor necrosis factor alpha, interleukin 6, interleukin 2, genes coding for inducible enzymes such as cyclooxygenase 2, inducible NO synthase, and adhesion molecules such as intercellular adhesion molecule and vascular cell adhesion molecules. Long-chain polyunsaturated fatty acids, namely, for instance, docosahexaenoic acid, stimulating the expression of inhibitory protein for nuclear factor kappa B, sequesters this nuclear factor in the cytoplasm, and signaling pathway leading to pro-inflammatory substances is interrupted. What about recommendations of long-chain polyunsaturated fatty acids N-3? From the epidemiological studies follows that in countries with high intake of these fatty acids, there is a substantially lower number of COVID-19 victims per 100,000 inhabitants in comparison with countries where intake of long-chain polyunsaturated fatty acids N-3 is low. Currently in European countries are recommended to intake 500 milligrams per day. American Heart Association recommends similar values, but Food and Drug Administration warns that it is not reasonable to exceed 3 grams per day of eicosapentaenoic and docosahexaenoic acid per day, it's meaning from all sources. A very strong source of eicosapentaenoic and docosahexaenoic acid is cod liver oil. 1 tablespoon delivers approximately 2 grams of these fatty acids, including 35 micrograms of vitamin D and also nearly 4 milligrams of vitamin A. So this source of long-chain polyunsaturated fatty acids is recommendable, but what is recommendable is natural consumption from fish And I would be afraid rather it's the same as I have told about zinc. Supplementation is questionable. From my side, it's all for this time. So I would like to thank you very much for your attention.
Thank you very much, Tomáš. It was very, very interesting. We can see some questions in the chat, but thanks to we are a little bit delay. Please let's answer for it at the end of our today's session. There is a question from Sarah about vitamin D3 and the vegans diet, and from Ivana there is a question on quercetin as a plant flavonoid. Or you can write the answer directly to the chat for the people who need to leave a little bit earlier. And Dr. Candida Rebelo is prepared as a third speaker. Now let's pass the balloon from the Mendel University to LSU, and Candida, the Meet Microsoft Teams is yours, please.
Thank you, Daniel. Good morning and good afternoon in the Czech Republic. I'm Candida Rebelo. I'm a research scientist and a registered dietitian at the Pennington Biomedical. In today's presentation, I'm going to talk about food as medicine in the context of COVID-19. So during the H1N1 pandemic of 2009, obesity for the first time was recognized as an independent risk factor for increased severity and death from an influenza infection. And as you heard earlier, obesity during SARS-CoV-2 has also been found to be an independent risk factor for increased severity of the disease, increased hospitalizations, increased ICU admissions, increased need for mechanical ventilation and mortality. In the United States, approximately 89% of people who have diabetes also have obesity. Diabetes and obesity are closely linked, and diabetes is also an independent risk factor for increased severity of a SARS-CoV-2 infection. So this figure plots BMI against age among patients hospitalized with COVID-2 infection, with the SARS-CoV-2 infection in the United States. So as you can see, as age increases, BMI actually goes down. But in younger patients, there is a higher need for hospitalization among patients with SARS-CoV-2. So obesity shifts the severity of the disease to younger ages and makes them vulnerable to the disease.
I'm sorry, Dr.
Candida, we cannot see your presentation. Something happened.
Oh, okay.
We can hear you very nice, but we
can you see it now?
Yes, we can.
Okay, so let us start here. So this was the graph I was referring to. In this graph, BMI is plotted against age.
It happened again. Sorry, it happened again. When you started to speak, the presentation was gone.
Now can you see it?
No, only you, your camera.
I'm sharing my screen.
No.
Can you try click? There we go. It looks like there we go. That's it.
Now can you see it?
Yes.
One moment.
And no.
And now we can't. It went away.
Now?
Nothing. Only your camera. Maybe please share directly presentation and not your desktop. Yes, we can see your presentation.
Can you see the presentation?
Yes.
Can you hear me?
Yes.
So now?
Yes.
You can see the presentation as well as hear me, right?
We can, yes.
Okay, so I'm going to start from here.
Yes.
Can you see this slide?
Figure, yes.
Okay, so as I said earlier, this is a slide. This is a graph that plots BMI against age. Can you still see it?
Nah, no. It's happened again.
I don't know. I'm not sure what to do.
Please use that arrow for sharing.
I did.
And share directly the presentation, not window, mean your screen, but the, yes, window, presentation window. Not the screen of the desktop.
Can you see it now?
Yes, we can see it.
And you can hear me?
Yes, perfectly.
Still, can you see me?
Yes, perfect.
Okay.
Please.
I'm going to skip this. I talked about it too much.
Perfect.
Okay.
We can see it. Leptin.
Okay, so is leptin the link? Leptin is a hormone that's released in proportion to body fat, and it regulates metabolism. Leptin also regulates immunity. So when a person is infected with the SARS-CoV-2 virus, these viral encoded products downregulate the interferon signaling system. It interferes with the interferon signaling system to downregulate the host antiviral response. So leptin plays an important role in these activated lymphocytes. Leptin acts through the JAK-STAT pathway to reduce T cell number and function. It regulates T cell number and function. In obesity,
the same problem.
He just come here 1 minute.
What happened?
They can't see my presentation. So this is it. I'm sharing my screen. Okay. So this is my presentation. But he's saying don't share the desktop, share the screen. Window.
Now something happening.
Dr. Rebello, that was the stop sharing screen button. So we saw you for a moment, and then once you click on that X, it goes away.
That. Yeah, but every time I click on it, and then this is my presentation, so I'm clicking on my presentation.
And right now we are actually not seeing the present. Okay, now it just popped up. It just popped up.
Okay, do you see me and the presentation?
We can see both you and the presentation, exactly.
Okay, but then it sort of goes away. Now?
We've still got you and the presentation.
Okay, I'm going to continue then.
Okay.
So, see, this is what happens. This is a screen that keeps popping up.
Okay, so it just went away. Your presentation just went away. So whatever you clicked on or whatever popped up, that made it go away. Now? Do you see me? I see you. I don't see the presentation at the moment.
That's the. don't know what to do. Do you want the next presenter to go on, and I'll just send somebody my slides?
If you can send slides, yes, we can definitely bring the presentation up on our end, and then we could just advance it for you.
Who do I send it to? To Ivana?
Yes, you
Yes,
can
ma'am.
send it to me, ma'am. I know you were a little time constrained.
Yeah.
Do you want us to move to the next speaker and figure this out?
Yes, go ahead.
Okay, thank you, Daniel.
Okay, thank you. So we will do a little bit of change. The next speaker who is prepared is Dr. Jacob May. What I have in my list of participants, or Dr. Yen Chen, but I've in the table schedule that the Dr. May should be prepared. So if you can share the presentation and continue with your topic, precision nutrition in the respiratory disease and lung function, perfect. We can see it.
Wonderful. Okay, and can you give me a quick reminder of what our time frame is here?
Pardon, once more, please.
Can you give me a quick reminder of what our time frame is? I can adjust the presentation to fit.
Yeah, you have that 15 plus 5 minutes, and now we cannot see your presentation. It was there only for a few seconds.
All right, I'm going to leave my video off and see if that does anything.
Now we can see. Yes, there is your presentation.
All right, you can see my presentation and an About Me slide?
Yes.
Okay, hopefully this will go smoothly. My pleasure to be here. I will move quickly just because I know we are a bit pressed for time. The title of my topic is Precision Nutrition in Respiratory Disease and Lung Function. So it's a little peripheral to COVID-19 specifically, but I'm sure you can all appreciate the symptoms of COVID being related to lung function. It's gone again, huh? All right, so I'm going to leave Teams and then come back, and perhaps that will fix the issue. But my guess is at this point that it may be a Teams related issue. So is it okay if I leave and then come back?
Okay, of course.
Yes.
Of course.
Just so you know, for those of you who are not aware, Teams did have some worldwide problems yesterday and the day before. We were lucky to not have encountered them in our first 2 days of our food symposium, but I guess we may be a little less lucky today. So here's what we're going to do. We're going to make another attempt to view the slides. If we cannot make that happen, perhaps we can have a little discussion with our scientists to give us a little overview as to what the topic they are working on is and try to create some discussion. We will then be putting all these presentations on our website together with the links and recording. So the link that I keep putting in our chat will have those presentations probably as of early next week. And we encourage you then to reach out to any of us and we can put you in touch with our presenters. But it looks like Dr. May is back on.
Okay, yes, we can
Yes.
try again. We can try again to get moving. So just a little bit about me before we move forward. I'll skip through my educational background and just mention that I'm a postdoctoral research fellow at Pennington Biomedical on a training grant to investigate botanical approaches to combat metabolic disease. And the research approach that I prefer to use is a clinical translational research approach, meaning we use clinical human trials in combination with cell culture and mechanistic studies to investigate the relationship between nutrition and disease. And then
we cannot see nothing again.
Boy, I am not sure at this point what may be going on. Let's see, it does just pop up on my screen. The screen share is just taken away. So maybe if there's a way to prevent other individuals from incidentally taking control when they highlight over Teams. I know GoToMeetings has a similar issue where if someone else has presenter capabilities and they move their mouse on the Teams screen, it transitions over. So my guess is you can see my slide again currently, correct?
Yes.
Yeah, so what I would ask is that anyone with presenter capabilities, just as a test, perhaps try to not touch your mouse at all, and let's see if we can go through this. And that is the perfect time to wish everyone a wonderful and happy St. Patrick's Day. I am going to just briefly review knowledge gaps in nutrition, metabolism, and respiratory function. I'll talk about 2 recent publications that we put out in the field, and then my potential interests and collaborative recommendations for precision nutrition in respiratory disease. So why am I interested in respiratory disease? Primarily because we don't have a pulmonary diet. And if you compare this to other organ level diseases like diabetes, cardiovascular disease, renal disease, we have some really great nutrition guidelines that we can give to people to say, hey, this will help control your blood sugar. This will help with your kidney function. We don't have that for pulmonary function and respiratory disease. And the COVID pandemic really highlighted our lack of knowledge in that area. There is some data out there in severe respiratory disease, for example, people that are admitted to the intensive care units where ketogenic formulas may be beneficial. This essentially reduces carbon dioxide production from nutrient utilization and eases the stress on respiration. However, there is just conflicting data on this, even on outcomes as important as mortality. And some studies looking at this have actually had to cut their study short because their intervention group actually had higher mortality. So it's really still an area that is much in need of research. There's also a common respiratory disease, which is asthma, which is a global health concern, and it reduces health span, increases hospital costs, and is estimated to impact nearly 400 million individuals by 2025. And again, we don't have standard recommendations for individuals with asthma. So first, I'm going to talk about some data that we generated in pulmonary arterial hypertension, which is a very severe pulmonary or respiratory disease. It's a rare disease characterized by pulmonary vascular resistance, which leads to right heart failure. And it has very poor clinical outcomes. Without treatment, the life expectancy is less than 3 years. And our understanding of nutrition and metabolic health in pulmonary hypertension is just really underwhelming, and there's a lot of new research in this area that I'm interested in. You can see the picture in the lower left corner here. This is the top of the stairs plop, so individuals with pulmonary arterial hypertension will really become exhausted or lose their breath after just going up something like a flight of stairs. So what we first did was conduct an oral glucose tolerance test in these individuals. And if you're unfamiliar with it, this is essentially a 75-gram sugar drink, and we give it to individuals and then measure items like their glucose or hormones that regulate glucose like insulin. And just to summarize this slide without diving too deep into it, is we found that individuals with this PAH disease have poor glucose control and reduced insulin resistance, and reduced insulin response to glucose. So they were actually similar to individuals with type 2 diabetes, as opposed to being more similar to otherwise healthy individuals, or maybe just individuals with obesity. So from this, that led us to want to do a more deeper look into the glucose metabolism in individuals with pulmonary arterial hypertension. So we designed a cross-sectional metabolic study where we performed a 3-hour hyperglycemic clamp. Now, with the oral glucose tolerance test, that's an oral beverage that individuals drink. With the hyperglycemic clamp, it's a little different. You've seen this image here of a patient laying down in a hospital bed, we actually infuse the glucose directly through an IV line. And from here, we can really measure how their body responds in terms of insulin, or we also measured C-peptide, which is a surrogate marker of insulin secretion. And this gives us a deeper understanding of metabolism. Now, I will mention this was a small study, but as you can see here, individuals were middle-aged and had a BMI in the obese range, but otherwise had normal fasting glucose levels and slightly elevated fasting insulin levels, but nothing that you would be concerned about in terms of type 2 diabetes. So just to summarize this study in pulmonary arterial hypertension, we found that individuals with pulmonary arterial hypertension, as I mentioned, have a reduced response to glucose, or they have poor glucose control when they consume exogenous glucose. And then when we infused glucose, we found they actually had alterations in insulin production, not so much secretion from the pancreas, but actually insulin is extracted from the liver, and it seemed like most of their regulation was coming from the liver. Now, we also looked at ketones and lipid oxidation by looking at the primary circulating ketone, which is beta-hydroxybutyrate, and also some measures like acylcarnitines, which are small metabolites in the blood that we can use to estimate lipid oxidation from. And what we found here was that individuals with PAH had a preference for using lipids and ketones as fuel sources. And we believe this is also in agreement with the information. When we give them exogenous glucose, they have a poor response to that. And we feel like this might be because their body really prefers to try and use ketones and lipids for fuels. Now, the thought is potentially, could this be a beneficial adaptation to their body trying to protect their respiratory function by trying to utilize items like ketones and lipids, which, as I had mentioned, kind of reduce the carbon dioxide or respiratory cost of energy, or is it just a measure of disease progression and metabolic disease? We don't really know, but it really brought up some interesting questions. As I mentioned, this aligns with the concept of the high-fat, low-carbohydrate pulmonary formulas that have been used clinically. But again, data is, we really don't fully understand what's going on there, but all those items seem to align. So I was then interested in looking at a more mild respiratory disease, like the very common asthma. So this was a separate study in individuals with and without asthma. A little larger than the last one, which, as I mentioned, only had 12 individuals. This we had 61 individuals, and we wanted to look at resting energy expenditure and its relationship with respiratory function and inflammation. And we also assessed habitual dietary intake, or dietary food preference in individuals with and without asthma. Our hypothesis was that energy expenditure would be increased in individuals with asthma due to the increased cost of breathing. So here's the participant characteristics. As you can see here, individuals were middle-aged, but relatively healthy blood pressure, resting heart rates. The items towards the bottom, if you can see the FVC, FEV1, and FeNO, these are all either spirometry measures or measures of airway inflammation and asthma. And as expected, the individuals with asthma either had significantly reduced lung function or trended that way based on the measure that we were looking at. Let me share my screen again. My guess is it popped off for you. Okay, can you all see my screen again?
Yes, we can.
Okay, beautiful. It seems to keep popping off. Mm-hmm. So again, okay. All right. Are we good? My apologies. I'm not sure what was going on there. It looked a little weird on my end, but we can see the screen.
Yes.
Perfect. Again, apologies for these technical issues. So when we look at resting energy expenditure, we compared measured resting energy expenditure, which we measured by indirect calorimetry, compared to estimated energy expenditure from common or clinically used equations that are based to essentially have us estimate energy expenditure or calorie needs in individuals in the clinical setting. And this is important because we base our nutrient recommendations off these estimates. And what we see here, compared to our control individuals, the individuals with asthma had elevated resting energy expenditure. In the field of energy expenditure, there's really been a lot of work done, and we've learned is that the best way to compare resting energy expenditure between groups is to really generate cohort-specific logistic regression modeling using 4 previously defined factors, that being age, sex, fat mass, and fat-free mass. And you can see that defined in model number 1 above. And what we do is we take that logistic regression model, and then we added various factors like asthma disease status or some of the measures of airway inflammation or other markers of immune function in individuals with asthma. And we tried to see if that would improve the regression modeling. And here what we showed is that the addition of asthma or FeNO, which is again a marker of airway inflammation, white blood cell count or neutrophil count, all improved that model and had an impact on the resting energy expenditure equation. So in this setting, essentially what this shows us is that individuals with asthma or individuals with biochemical measures that are consistent with the asthma phenotype, all related to having elevated resting energy expenditure. We also, as I mentioned, look at habitual diet in these individuals. And here we showed that individuals with asthma report consuming less carbohydrates than their healthy counterparts. Although fat intake and protein intake trended to be a little different as well, those were not significant. The primary significant difference was in that carbohydrate intake. And this again is in line with our primary hypothesis from the pulmonary arterial hypertensive work that perhaps the body is trying to self-regulate and improve respiratory function on its own. And I think that's really an area that we can further investigate, is whether or not this reduction in carbohydrates or potential shift in fuel preference towards lipids and ketones may have an impact on lung function and potentially an application in respiratory disease. But we certainly do have a long way to go until we can get to that conclusive point. These are just some early data and publications that we've recently put out, and I just wanted to describe sort of my thought process and where I'm hopeful that this research can go. One of the other potential reasons for this difference we see in fuel preference could be due to bioenergetic alterations. And this can go as deep down as the mitochondria in the way that our bodies utilize fuel for energy. So I'm going to share a little data from an in vitro study that we did. This was again also recently published in a different context. Here we looked at human skeletal muscle cells and we assessed mitochondrial respiration using the seahorse equipment. And essentially what we did here is we put different doses of ketones in with the nutrients that we fed our cells. And we found that low doses of ketones actually had a significant effect on mitochondrial respiration, whereas higher doses didn't seem to have an effect. And so this kind of fueled my thoughts on, whether or not there's an optimal dose of ketones that may be beneficial for certain areas of health like metabolic disease or respiratory function. I do want to note, I didn't present this data here, but in our individuals with pulmonary arterial hypertension, they did have elevated circulating ketones. And now the amount of elevation that they had was actually quite small. So if you think of ketones in millimolar concentrations, typically, if you have a healthy person after an overnight fast, especially if it's a longer one, they'll have roughly 0.5 millimolar of ketones circulating around. If you do something like the ketogenic diet, you have about threefold that amount, 1.5 millimolars. And I want to mention that all the individuals we studied in this report actually had much less than even the healthy levels. So my current thought is that it's not necessarily an elevation of ketones that may show benefit in these different areas, but rather just a return to normal ketone levels, and whether or not these ketone levels are reduced in various diseases is really something of interest to me. And finally, I want to talk a little bit just about opportunities for precision nutrition and the sort of research that I'm really interested in doing. And precision nutrition is really a combination of multiple approaches to allow us to target nutrition advice towards the individual. So taking a lot more into account than just population averages. But to get there, we do still have to use the large-scale epidemiologic approaches. So I'm doing work with our data set here in the United States, which is the NHANES, which is essentially our largest nutrition and health survey that we have. But I'm very interested in looking at large-scale epidemiologic relationships between lung function and dietary habits. So certainly interested, to chat with anyone working in that area. In terms of the inpatient trials that we do, I definitely would be interested in looking at different feeding trials and how that relates to lung function or something like asthma symptomology. And then finally, the biological mechanism piece, which is that cell culture work. I had mentioned we had looked at whole body fuel preference, but there's also this information that I've shown to you about the mitochondrial bioenergetics. So certainly anyone doing work in that realm regarding mitochondrial function, especially in cells that are relevant to respiratory disease, like airway, smooth muscle cells, or otherwise. And so our first goal with this would be really to evidence the best nutrition to support lung function. And then the second goal is to utilize the precision nutrition approach, which is using omics-based approaches like genomics, proteomics, metabolomics, and use machine learning and algorithms to generate precision nutrition recommendations so we can have an individual come in, phenotype them, genotype them, and say, you know what, we think this nutrition is best to help you with your current clinical issues. So those are certainly lofty and long-term goals, but that is just the area that I am interested in. So in closing, the role of nutrition and metabolism and lung function really has yet to be fully elucidated. I think there's a lot of potential there. And I just want to reiterate a message that Candida put out there, which is, we really do believe that food is medicine, and we can utilize precision nutrition approaches to improve our treatment and management of disease, especially in combination with pharmacologic approaches. And with that, I'll just show my acknowledgements. And the final slide to connect with us at Pennington Biomedical down here. I do highly recommend all health and nutrition professionals are on Twitter. You can see my tag there as well. It's really important for us to get out there and put out good information just to really combat all the misinformation that we see. So if you're a health professional interested in getting on Twitter, please contact me, let me know. I'd be happy to guide you in that direction. And with that, I'd be happy to either take questions now, or if we want to move forward with the presentations, I'll hang on.
Thank you very much. It was very interesting. The people who are obese was, let's say, warned a little bit at the previous presentation. Now the people with asthma, maybe it's beneficial for them to have asthma. I only let's say didn't understand the equilibrium between asthma and, let's say, diet with the low carbohydrates and the ketones. So because usually the people who don't eat the carbohydrates has a more ketones in the blood. So can you explain for not so educated people like me how it works, or if it's better to have that ketones in the blood, let's say, against the COVID?
Yeah, so that's a great question. I think in the fasted state, like an overnight fast or when you haven't eaten food for a little while, you really do want ketones to increase and be a good fuel source for you. In terms of the dietary changes that we saw, to really initiate a significant change in ketone levels, you have to drastically change your carbohydrate intake. So I don't think our individuals with asthma changed their carbohydrate levels enough that it would have an impact on their ketone levels, but I am interested to look at the physiology of asthma and whether or not there is anything internal that is driving these individuals to eat less carbohydrates, maybe towards that approach. Maybe they're attempting to have something internal that would improve ketone utilization, and part of that involves reducing carbohydrates. But we really don't know just yet. And I will say, typically, the amount of carbohydrates you'd have to reduce to significantly increase ketone levels is probably about 90% of your carbohydrates. The difference we saw here between our control group and our asthma group was only about 15 to 20%. So I wouldn't expect it to have an impact on ketone levels, but I do think the physiology may be underlying there to have an impact in that area. Not sure just yet. Great question, though.
Thank you very much. I have information from Louisiana that Dr. Chen has apologized for leaving us. She had to go and had some duties, and her presentation will be recorded and added to the overall record of our session. And if we can try again, Dr. Rebello, your presentation, please. Let's do that.
Okay, I do have Dr. Rebello's presentation, so let me try that. And I would appreciate a feedback if you see anything.
Something happened?
Something is happening.
Looks good. Yes, we can see it.
Okay. Are we good with this view, or do I need to do anything?
Looks good.
Perfect.
Thank you. Dr. Rebello, it's all yours. There you go.
Okay, I think I went over all this. I'm not going to start all over. Maybe I should just go from that. Did you see? Y'all didn't see the slides, or should I just start from scratch?
We can see first slide, and the leader of the presentation is Ivana. So Ivana has to move the presentation.
Just tell me what you want me to do and I will move it for you.
Okay, so I'm Candida Rebello. I'm a registered dietitian and I'm a research scientist at the Pennington Biomedical. In today's presentation, I'm going to talk about food as medicine in the context of COVID-19. So you all heard about how obesity increases the severity of a SARS-CoV-2 infection. I'm not going to labor this point. I talked about it earlier. Let's move on to the next slide. So in this slide, BMI is plotted against age, and it shows that at younger ages, people who are hospitalized with COVID-19 or the SARS-CoV-2 infection have a greater risk for being hospitalized in the United States. So it shifts the severity of the disease to younger ages. The next slide. So is leptin the link? This was a paper I published earlier in 2020, suggesting that leptin might be the link. So when patients are infected with the SARS-CoV-2 virus, the viral encoded products interfere with the interferon signaling pathway and they downregulate the host antiviral response. Leptin is a hormone that's released by adipose tissue and it is released in proportion to body fat. In obesity, leptin is dysregulated. Now, leptin regulates metabolism and leptin also has a role in immunity. The dysregulation in obesity arises from a condition of leptin resistance. And in these patients, there's high circulating leptin. So high baseline leptins result in immune defects. There is a condition called lymphopenia that has been a consistent finding in SARS-CoV-2. This has led to tissue damage, increased release of inflammatory cytokines by macrophages, and an uncontrolled loop of self-amplification that results in some patients in multi-organ failure and death. So this paper proposed that leptin may be the link between SARS-CoV-2 infection and obesity and increased severity when someone has obesity. So the next slide. The next slide. At this time, there are no, not this one, the previous one. At this time, there are no drugs to reduce leptin levels. One way to reduce leptin levels is to reduce body fat, because leptin is released in proportion to body fat. I have been working with food components, and I look at the effects of food components on energy expenditure. Energy, when people lose weight, lifestyle approaches are recommended as the first line of treatment. And when people lose weight, they decrease energy expenditure. After a while, if that decrease in energy expenditure continues during the weight maintenance phase, it becomes very difficult for them to maintain that weight loss. So it results in a cycle of weight loss and weight regain. And for most people, that's very discouraging. Any strategy to increase energy expenditure is a good adjunct to lifestyle approaches. And that is what my research is focused on. I look at food components and I work with human adipose-derived stem cells. So we get adipose tissue from patients who are having elective surgery. We extract the stem cells and then differentiate them into adipocytes, treat them, and look at the effects of food components. I have been working with naringenin, which is a citrus flavonoid. I use an extract of whole sweet oranges that has been standardized, quantified and standardized to 30% of naringenin. Naringenin is a PPAR gamma and a PPAR alpha activator. So when PPAR alpha and or PPAR gamma are activated, they induce the expression of genes that are related to thermogenesis, like UCP1 and the others I've listed there. And these are highly thermogenic. That means what UCP1 does is it short circuits the mitochondrial energy, the proton gradient, which results in the release of energy as heat. So it's highly thermogenic. It leads to an increase in energy expenditure. The other genes involved are also the ones related to glucose metabolism that are upregulated by naringenin. The next slide. So in this study, we showed that in these human adipose-derived stem cells that are differentiated and treated with naringenin, there's an upregulation of UCP1 and CPT1B, which is a key enzyme that's involved in fat oxidation. So if you're going to have a thermogenic procedure, you need the fuel for this process, and CPT1B is important for providing fat oxidation. When we inhibit it with using either PPAR alpha or PPAR gamma inhibitors, UCP1 and CPT1B is downregulated. So clearly, naringenin is acting through these PPAR transcription factors, which are ligand-operated transcription factors. The next slide. In human white fat, in human white adipose tissue, as well as in human adipocytes, we showed that naringenin upregulates UCP1, CPT1B, GLUT4, which is the glucose transporter, and carbohydrate responsive element binding protein, which is a key enzyme that's involved in insulin sensitivity. So this was both at the messenger RNA as well as the protein level. The next slide. It's all very well when something works in cells, but if it cannot reach the cells, it cannot do its job. I then conducted a clinical safety and pharmacokinetic study to show the bioavailability of naringenin in humans. I tested 4 doses, 150, 300, 600, and 900 milligram doses of naringenin. At the 150 and 600 milligram doses, following ingestion of the dose, I measured naringenin concentrations in serum over 24 hours. At the 300 and 900 milligram doses, I measured it over at baseline, 4 hours and 24 hours, because 4 hours was estimated to be the optimum or the peak concentration. And as you can see, at 24 hours, it's no longer present in circulation, but the peak concentrations are approximately 40 micromolar. We use it at 8 micromolar on the cells. So clearly, it's highly bioavailable. Also, there were no safety concerns. The next slide. I then conducted a case study in an African-American female subject. She had obesity and a diagnosis of diabetes. She was prescribed metformin, but she had stopped taking it because metformin, as it does with many people, gave her adverse gastrointestinal effects. We gave her the lowest dose, 150 milligrams, because we didn't have the pharmacokinetics data at the time of the case study. So with the lowest dose, 3 times a day for 8 weeks, no restrictions on her food intake. She was told not to change her lifestyle. She still lost 2.3 kilograms in 8 weeks, and her fasting insulin reduced by 18%. There was an increase in her metabolic rate, a small increase in her metabolic rate. The next slide. I then looked for compounds or food components that would synergize with naringenin in order to enhance its effect. And we tested several components and found that the carotenoids act in synergy with naringenin, particularly beta-carotene. Now, beta-carotene is the precursor of vitamin A, and it's the major form of vitamin A in the human diet. Beta-carotene is found in foods like sweet potatoes, mangoes, pumpkin, orange-colored fruits and vegetables. Beta-carotene is an RXR, retinoid X receptor agonist. The retinoid X receptor forms a heterodimer with PPAR gamma. And when both of them are bound by their respective ligands, they act in synergy. And that's exactly what we see. Naringenin and beta-carotene together upregulate UCP1 and GLUT4 to a much higher extent than naringenin alone. Similarly with ATGL, which is the enzyme that's required to break down triglycerides. Adiponectin is a key enzyme involved in insulin sensitivity, and adiponectin is also significantly increased with the combination. So these were seen both at the messenger RNA as well as the protein level. Next slide. So here we showed that PPAR gamma is upregulated not at the messenger RNA level, it's upregulated at the protein level, which is key. We want an upregulation of PPAR gamma protein, and even its co-activator PTC1 alpha was upregulated at the protein but not at the messenger RNA level. The next slide. I then conducted messenger RNA sequencing of human adipocytes that were treated with the combination of naringenin and beta-carotene. So this messenger RNA sequencing and the bioinformatics analysis revealed a host of secreted factors from adipose tissue that are involved in energy expenditure and glucose metabolism, as well as the genes in involved in thermogenesis, such as UCP1 and others. Many of these that have never been characterized in human white adipose tissue, and there were no antibodies to check, to corroborate the evidence from the bioinformatics analysis. But we were able to validate some of the information through RT-PCR. And I'll tell you about another case study that I'm going to be doing in order to find out what happens with circulating proteins. So the next slide. These are 2 thermogenic agents that were upregulated in the bioinformatics analysis, and we confirmed it through RT-PCR, PM20D1 and CKMT1. Next slide. PM20D1 is activated by PPAR gamma only in humans, not in rodents. And it is the enzyme that is responsible for synthesizing these N-acyl amino acids. N-acyl amino acids are endogenous uncoupling agents. They do exactly what UCP1 does to increase thermogenesis and energy expenditure. And they do it independently of UCP1, regardless of whether the cell is UCP1 positive or negative. So it's a highly thermogenic process. CKMT1 is an enzyme that transfers a phosphate group from ATP to creatinine to form creatinine phosphate. That releases ADP for ATP synthesis. Again, this is also a highly thermogenic process. So these were genes that we discovered through the bioinformatics analysis that we were able to confirm through RT-PCR. Next slide. I'm now going to do another case study in the same subject who did the earlier case study. But this time we will treat her with naringenin and beta-carotene, and naringenin at the high dose of 900 milligrams, which we tested and found to be safe in the pharmacokinetics clinical trial. So she will be given naringenin and beta-carotene for 8 weeks. Prior to starting the supplementation, we'll measure weight, resting metabolic rate, oral glucose tolerance test with glucose and insulin. For safety measures, we'll look at 24-hour ambulatory blood pressure. We'll draw blood for a complete blood count and a chemistry panel as safety measures. And we will draw blood, and the serum will be used for a proteomics analysis to evaluate what is released into circulation for the proteins that we could not find antibodies, since they are not characterized in humans. 24 hours post the naringenin and beta-carotene dose, we will draw blood again for proteomics to determine what are the factors that are released acutely following the dose. She will continue to take the naringenin and beta-carotene for 8 weeks. At 4 weeks, we do a compliance check and a weight check, a blood pressure pulse, and then at 8 weeks, we repeat all of the baseline measures. So I'm hoping that the proteomics analysis will give us an insight into what are the secreted factors by adipokines. The bioinformatics analysis, for instance, showed RBP4, which is downregulated, and that's what we would like to see, particularly in subjects who have obesity. The next slide. The only other food component that's been shown to reduce leptin is curcumin. There's a lot of data in the literature in support of curcumin, and earlier it was found to be not bioavailable in humans, but now it's being delivered with piperine, which enhances its bioavailability. In human trials, it's been shown to be tolerated at doses ranging from 500 milligrams to 8,000 milligrams a day. Both animal as well as human trials have shown clinical benefits in respiratory inflammatory diseases with curcumin supplementation. In 2 clinical trials, curcumin has been shown to reduce leptin levels independent of weight loss. One study demonstrated it in 7 days, and the other study demonstrated a reduction in curcumin over 8 weeks. Next slide. And so in summary, obesity increases the risk of severity in SARS-CoV-2. Leptin is elevated in obesity. It links nutritional status with the immune responses. So treating obesity is preventive not just for chronic disease, but also for infectious disease. And if we let the disparities that COVID-19 highlighted persist, we will likely have a similar toll during the next pandemic. Naringenin and beta-carotene increase energy expenditure safely. Naringenin and beta-carotene are a PPAR gamma activator. There are PPAR gamma activators on the market called TZDs for the treatment of diabetes. A number of them have had to be removed from the market for adverse effects, and even the ones that are on the market produce weight gain. Naringenin and beta-carotene increase energy expenditure safely because they are targeted, and now there are many selective PPAR gamma modulators that are an active area of drug research. However, naringenin and beta-carotene do that, and they are natural products. Curcumin reduces leptin independent of body weight. Its effects in SARS-CoV-2 remain to be established. So foods with safety and efficacy profiles must be rigorously investigated, like we have done. We've gone from cells to human, back to cells, and we still have work to do before we can take it into an efficacy clinical trial. Next slide. And that's all I have today. For today, my acknowledgements. My primary mentor is Dr. John Corbett. Dr. Frank Greenway runs the lab, which tests food components. Ann Coulter is the director of the lab. Guru Ramanathan and Raul Manalak are the other members of the lab. I'm funded through a K99 ROO from the National Institute of Aging, and some of this research was funded rather by the Brown Foundation. Thank you.
Thank you very, very much, Dr. Rebello, for your very, very interesting and nice presentation. I'm very glad that we finally could see it. You have a question from Dr. Matejovichova. Does it mean that the pre-adipocytes are turned via naringenin into so-called beige adipocytes?
Yes, we think that it does cause a beiging of white adipocytes. That's why there's an increase in energy expenditure. So it does do a beiging of white adipose tissue.
Mm-hmm. Thank you very much. If I can see the questions what we didn't answer, so you as a presenter speakers, do you have a question to each other? I did.
May I ask a question to Candida?
Yes.
Of course.
Wonderful. Great presentation, by the way. I'm curious your thoughts on the naringenin and whether you think naringenin or even minor, I don't even want to call them nutrients, but minor phytochemicals like that will ever have a role in dietary recommendations, or if it's always going to be towards building something like a supplement or working towards a pharmacologic compound.
That's what we are headed towards. Unless we can show randomized controlled trials with efficacy, that's when it can be taken. That's when recommendations can be made, as dietary recommendations. Because these are, if this was a drug, it would have gone through another 10 years of drug trials. But these are safe products. They don't go to, and that's the reason why they can be cheaply available, not like drugs, and we can make recommendations for them. But we have to go through a process, a rigorous process to establish their clinical safety and efficacy, which is what I'm trying to do.
Perfect. Thank you.
If I may have a question, please, for all of our panelists. Can you let us know what your international collaboration ideal would be? What type of collaboration are you looking for, and what is the outcome? Is it publications? Is it, in some of our presenters, it could be some student events. So if you can just touch up on that a little bit.
I can jump in and start. So at least from my interests and the role of precision nutrition, I think a key area is even just performing the same sort of experiments or outcomes in different populations, and being able to really just increase the total, what you could call maybe database of information that we have. Precision nutrition really relies on having a lot of information, as I had mentioned, those omics-based approaches. And I think the more diverse we can make our populations, whether that's something as different as location or just habitual dietary differences, I think that's really beneficial. So anything from the human realm that is even just conducted in different populations, I think it's very beneficial.
Yeah, I agree with Jake. If there's anyone who's interested in looking at food components, if you have ideas or you have a population that you'd like to test things out in, sometimes it's very expensive to do clinical trials in the United States because it's very, very expensive. So maybe it's possible to do it elsewhere with different and diverse populations, or even to test these food components, in other populations.
Dr. Compra.
From my point of view, we view you in you in research institute have much better position than than we, because we teaching nutrition to students, but regarding experiments in cell cultures or humans, it is because the Mendel University is from its character, it's agriculture university. So some experiments on in humans, it's very difficult for now. We are able to do some animal models in rats and mice, but experiments in humans will be very difficult for us. So, for instance, for our students, because we are teaching human nutrition in Mendel, but our students have difficulties to perform their praxis in clinical departments. It's not easy for us. So we are rather pressed to do experiments in animals and in cell cultures only.
Thank you. I will give a space to Dr. Mateovichova if she would like to say something before the questions, what we can see in the chat.
Okay, thank you for letting me also make my some expression. I think it would be very nice to have opportunity for students, for example, for PhD students, to cooperate in your labs, for example, on their studies or their topics, or exchange of students between our universities, because then it means more opportunities for topics of PhD studies. We can choose. That's I agree with my colleague. That's a problem, that we can't do any research on the human models or on the human, and it will be very nice if we can choose topic where the research on human can be done somewhere else, for example, in your place, and we can, on the other hand, take, for example, students from your university and teach them how to do analysis of food, and we have some pilot plants where they can try to do some work in recipes, and even the hazard, for example, hazard area, hazard and safety food area. So if there is some possibility in such type of cooperation, it will be very nice.
Thank you very much. We have a question about the curcumin. Maybe Dr. Rebello, it's for you. You spoke about it.
I don't work with curcumin. I was quoting things that are in the literature, but from what I can tell, that black pepper enhances its absorption. So that's why it's delivered along with piperine now.
Sorry, I had some interruption in internet, but I'm back. Thank you. Hope that everybody could hear it. In my overview, what I did for myself, that we heard about obesity from Dr. Matejevičová and Dr. Rebelo about the leptin, human fat prevention, beta-carotene and naringenin from grapefruit, curcumin. Then it was a very nice topic about carbohydrates in diet and level of ketones from Dr. May. And together with importance of vitamin D and zinc from Dr. Komperda, so it goes very nice together. Unfortunately, we didn't hear a presentation from Dr. Chen, and it was planned to be about the tea, so I hope we will see it from the recorded version. Because everyone wants to increase the immunity, and the presentation, her presentation should provide for us the overview of the prior research and nutrient value of the tea. So all together, maybe if I can see the Tomáš Komperda here again, there was a question about the pills, means the supplements of the vitamin D3. You said it's a little bit dangerous, but I had a joke on the Americans that they have at the breakfast every morning the full hand of the pills. No, I'm joking. And do you think it's okay? And there was also a question: what about the vegans? Can the vegans have enough of zinc and vitamin D? From the normal diet without of supplements? Thank you.
Yes, and I can to answer this question. I have to admit that I am not very friendly. I have not very friendly attitude to supplements. I would prefer natural foods. Regarding vegans, I am afraid that vitamin D will be not sufficiently present in their foods. I am afraid, because vitamin D, for instance, in legumes, in cereals, though these cereals will be whole corn, there is the level of vitamin D is zero. So if only one source of vitamin D which is not from animal sources are mushrooms. But it's very seasonal matter. So in summer season, also vegans, I think, will have sufficient vitamin D because in summer season there is, I hope, plenty of UV radiation from the sun. But in winter, I'm afraid that vegans will be very bad about supplementing vitamin D. Regarding zinc. Zinc, contrary to vitamin D, is also present in legumes, whole corn cereals, but I'm afraid that it will not be sufficient source for vegans. If I mean vegans, strict vegetarians, so they are against all meats. Also, they don't eat milk products, fish. There will be carency not only in vitamin D and zinc, but especially, I'm afraid, in iron. And regarding vitamins, especially vitamin B12, which is present in animal products. So I don't know how, because I repeat myself, but I'm not very fond of vegan nutrition, and I don't know how these vegans balance their diet regarding these some vitamins and minerals, especially. On the other hand, I admit that vegans have lower plasma lipid levels. It's okay, but regarding minerals and some vitamins, I would be very skeptical.
Thank you very much for the comment and answer. Thank you. I cannot see any question in the chat, so I will thank you to all the participants.
Excuse me there, but I have chatted
Okay.
for Milena after her first very interesting presentation. May I ask?
Of course.
Or it's not time enough, and we can discuss it in person.
If you think it's important also for the other participants, then yes, definitely.
Yes, Milena, once more, thank you for your very interesting presentation. You have mentioned orexigenic and anorexigenic signaling in hypothalamus, and as far as I know, there is different signaling when one consumes highly processed foods, contrary to so-called natural foods. It is some different signaling regarding oxygenic, anoxygenic signaling, because it is follows from epidemiological studies that when people consume highly processed foods in the same amount, the same energy content, it's different signaling in hypothalamus.
Yes, that's true. That highly energy content food has increased the orexigenic signaling, and it might be through different content of fatty acids. They are in saturated, more prevalent in saturated fatty acids than in unsaturated and polyunsaturated fatty acids. That's what did you mean, I think.
Yes. Okay, thank you.
Perfect. Thank you very much for your answer.
Dan, excuse me, I see one more question from Ivana here. There is some recommendation for supplement quercetin.
Yes.
Are you familiar with this, and do you recommend it?
Do you know it?
No, my answer is similar to my previous one. I do not like the word supplementing. So quercetin is maybe have something to say, Dr. Candida Rebelo, because quercetin is similar, is from similar group of substances as naringin. The quercetin is naturally present in berries, for instance, blueberries, strawberries, black currant, red currant. So I would recommend consumption of these foods, but not as a supplement.
Thank you.
Between the carbohydrates being okay and getting the berries, my diet is now full and complete.
And one more question, it's possible that done for Dr. Candida Rebello.
I think she's not here. She had to leave.
Not present. Okay, I'm sorry, because she has mentioned intermittently naringin and naringenin, and my question was if aglycon and glycoside functions similarly, or is there a great difference? Oh, sorry, never mind.
You can write to her the question by email, because you as a presenter are now deeply connected. You know about each other, so we can, of course, and we will continue with the collaboration. So
thank you. Thank you.
Thank you very much. If there is no question, I would like to thank you to everybody, especially to our presenters. Your presentations were very, very nice and in a high quality. To the participants, thank you very much for your patience, especially when we had technical problems. And I would like to invite you for tomorrow, where will be the technology or food technology block, what will begin at 8 o'clock in the US time and at 2 p.m. in the Czech Republic. So you are very welcome, and have a nice day, afternoon or evening. And it was very nice to spend the time with you. Thank you. Goodbye.
Thank you.
Thank you. Have a great day and evening.
Thank you also for the opportunity. Goodbye.
Bye.
Bye-bye.